Welcome everyone. My name is Ben Kaler. I'm the Science Coordinator for the Gulf Coast Prairie LCC. The Gulf Coast Prairie LCC is a collaborative science support partnership working to deliver sustainable cultural resource conservation in the Gulf Coast Prairie geography by sharing scientific knowledge, leveraging resources, and working towards common landscape goals. Today, we're going to have a presentation for our December lunchtime webinar series 2016 on an LCC project we're funding for Guadalupe Bass. So just a little bit of background about that species. So the LCC chose 28 focal species that represent 17 broadly defined habitats in order to define, design, and deliver long-term landscape conservation to achieve the mission of the partnership. And in August of 2014, our steering committee approved a science strategy which identified the most critical science needs for 6 of these focal species. And one of them is the Guadalupe bass. We chose that species to represent several aquatic habitat types. I just want to remind everyone that we're in listen-in mode right now, which means that everyone's muted. We're going to unmute everyone at the end of the presentation for open discussion, so just jot down some notes or questions in anticipation of open discussion. So today we're joined by Heather Williams, a graduate research assistant at the Department of Natural Resource Management at Texas Tech University in Lubbock. In just a moment, she's going to present to us an update on an LCC-funded project focused on understanding how in-stream flow affects growth and recruitment of Guadalupe bass. Before she takes the floor, I just want to briefly introduce her. So Heather grew up on the Guadalupe River in New Braunfels, Texas, where her love for freshwater systems began. Heather is completing a Master's of Science at Texas Tech. Passionate about freshwater conservation, her work centers on flow effects on growth of Guadalupe bass in North and South Llano River systems in central Texas. Her research interests include aquatic ecology, entomology, restoration ecology, and river conservation. Also, Dr. Robin Verbal-Pearson, an assistant professor at Texas Tech University, has also been heavily involved in these efforts, serving as a principal investigator at Texas Tech. However, she's unfortunately unable to join us today as she's attending to a family emergency. She completed her Bachelor of Science in Biophysics from University of Southern Indiana, her Master's of Science in Forest Entomology from the University of Arkansas, and a PhD in Applied Ecology from the University of Arkansas at Little Rock. Dr. Verbal-Pearson's research focuses on insect responses to disturbance in North American systems, and although we know her absence, we acknowledge her role in the effort at hand. So without further ado, Heather, we welcome you. You have control of the webinar and the floor is yours.
Thank you, Ben. So this is actually my first chance to speak publicly about Guadalupe bass, so I really appreciate the opportunity. A shout out to Tim Grabowski. He was the one who originally wrote the grant to fund this GCP LCC funded research. Quick outline, we're going to give a brief species introduction and then talk about the flow ecology hypotheses related to Guadalupe bass growth and recruitment, discuss previous research on flow ecology of Guadalupe bass, and discuss my project proposal and a request for sample. So like other black bass species, Guadalupe bass are not true bass. They're in the Centrarchidae family. It is the official state water fish of Texas and endemic to Central Texas. They're considered ecologically and economically important. They have this great potential as an effective indicator species of stream health due to their endemic nature, their high trophic level, and specific habitat requirements. They are estimated to generate $71.5 million in economic activity done by this recent really great research into the economy of Guadalupe bass, and including 776 full-time jobs created by Guadalupe bass. So they have this potential as a great charismatic and iconic symbol for both water conservation and watershed management in the state of Texas. So this distribution map was created by selecting subbasins that contained Guadalupe Bass occurrence points from the Fishes of Texas database. This included 365 occurrence points, and they ranged from 1938 to 2008. So Guadalupe Bass are endemic to the streams and rivers draining the Edwards Plateau. So subbasins found throughout the Brazos, Colorado, Guadalupe, and San Antonio River basins, and there's an introduced population in the Nueces River basin. These three photos show an example of stream habitats where Guadalupe bass were found during our 2016 field season. They're associated with spring-fed streams with clear to moderate turbidity and high current velocities, associated often with riffle habitats. But we also find them often in these habitat transition zones from riffle to run or run to pool. But Guadalupe bass are thought to represent a riffle run dependent guild of species requiring a relatively undisturbed mosaic of stream habitats. So discussing just the conservation status, threats, and current action, Guadalupe bass is listed as a species of conservation need by the state of Texas, and this is due to decreased stream flow, habitat degradation, and specifically introgression with introduced populations of smallmouth bass throughout their range. This decreased stream flow and habitat quality is mainly due to human activities and population growth, and thus are likely to continue into the future as Guadalupe bass occur in some of the fastest growing counties in the state of Texas. The Guadalupe Bass Restoration Initiative by Texas Parks and Wildlife is this holistic community-based approach to healthy watersheds, and this is done through landowner incentives for promoting healthy watersheds and riparian areas and decreasing invasive species as well. This has also included stocking of genetically pure Guadalupe bass to try and increase the genetically pure population from the threats of the smallmouth bass introduction. And as Ben stated, the Guadalupe bass are recognized as one of the GCP-LCC's focal species, and that came of light in 2014. So it's one of the 28 species identified, and they represent an indicator for river and stream health. So in 2015, the GCP-LCC partnered with SARP to create these flow ecology hypotheses. They're regional hypotheses for the ecological response to flow alteration. And there's these representative species throughout, like fish, bird species, and riparian vegetation that are thought to be affected for these different flow metrics. And so Guadalupe bass represented a guild of riffle run dependent species. And so 3 hypotheses were proposed by Tim Grabowski in this 2015 report to outline the flow ecology hypothesis. However, the data available, there hasn't been data available to test and refine many of these hypotheses, not available. Just going over them quickly, the first hypothesis is that Guadalupe bass growth and ultimately recruitment would be negatively correlated with increased magnitude and duration of extreme low flow events. This is because extreme low flow events can alter the availability of the riffle run complexes that Guadalupe bass are thought to be dependent on. The second hypothesis is that Guadalupe bass habitat quality and availability is negatively associated with both reduced base flow and magnitude and duration of extreme high flow events. So the quality of and availability of habitat for Guadalupe bass is thought to increase with increasing flow, but then at some point the flow becomes too high and could then reduce the quality or availability because it takes more energy to maintain their position. So this is in relation to flow. Guadalupe bass, they're also strongly associated with the riffle run habitats, and it may be due to drifting insects, because they rely on them more than other Micropterous species for prey items, which could be affected then by this base flow magnitude. The final hypothesis is that nest success of Guadalupe bass is negatively correlated to the duration and magnitude of high flow pulses during the spawning season. Like other shrimp tarkids, Guadalupe bass build and defend nests while spawning. And so when the high pulse flow gets too high, these areas could get washed out. And reproductive success may increase with decreased high flow pulse during the nesting season, which is March through June. So I want to highlight some of the flow ecology studies that have been done here at Texas Tech. Grushel, her Guadalupe Bass research mostly focused on habitat association, but she did do a growth analysis in relation to flow, and these collections were done specifically on the South Atlanta River, and she found a negative correlation between low discharge and growth of age 0 Guadalupe Bass, and also low recruitment during period of low discharge. So this kind of connected with the first hypothesis in relation to extreme low flow events. Mashur 2016, so he incorporated both the North and South Llano for 12 different representative species, and the Guadalupe Bass was one of them, and so they were captured on the North Llano and South Llano from 2014 to 2015, and growth was shown to be positively correlated to both PC1 and PC2. So these maximum flows were positively correlated with growth for all standardized ages of the Guadalupe Bass captured. And then looking just at the standardized growth during the first year of growth, captured the same place, North and South Llano River. Growth during the first year of life was also positively correlated to the PC1 and independent of river origin. The first year of growth was shown to have higher variability compared with standardized growth for all ages. Yeah. So now my current project objective from this project that is funded by the GCPLCC is to continue monitoring the effect of flow on Guadalupe Bass growth and recruitment throughout their range. No research has been done on age and growth relative to flow in basins outside of the Colorado River Basin. And the second component evaluates age 0 Guadalupe Bass at a relatively fine spatial and temporal scale to assess the mechanisms by which flow conditions impact age 0 Guadalupe Bass. So here I'm doing daily growth for the first time. All the other age 0 studies were done with relative growth or just growth within that whole year when those ages were back calculated. But here we're looking at daily growth on the North and South Llano River again. So a project for the age 0, it's again established on the North Llano and South Llano River, and sampling is done every 10 days, June through August. We hit at least 4 sites each of those 10 days, so it goes to a total of 8 events. And 8 events and 8 sites per event. The sampling is standardized, 1,000 seconds of backpack electrofishing and 6 seine hauls. The first 10 young of year Guadalupe Bass at each location are euthanized and retained for growth analysis and stomach content analysis, and then the remaining individuals are measured to the nearest millimeter and released. So the North and South Llano River are a good representation because as sister rivers, they have a lot in common, but they have considerably different stream flow, with the South seeking to retain a moderately consistent stream flow and the North Llano having more human impacts withdrawals. So I'm looking at daily growth for each 10 days. This box plot just shows the total length for the different events captured throughout the summer. The 2015 field season, we had a total of 402 young of year Guadalupe Bass. In 2016, we had 662. Those are currently being, the otoliths are being examined for both age and growth in the last 10 days. And so that will then be related through a principle, through my mixed models in ANCOVA to relate the that daily growth within the discharge between these 2 rivers. I'm also analyzing stomach content between all the young of year, and seeing it's interesting because then towards the end of the summer they start to switch to piscivory, but then in the beginning seeing the effect of insect drift on prey items selected by Guadalupe Bass young of year. So for my range-wide assessment of flow, We have 107 range-wide sites established in portions of the Brazos, not the Colorado, just the Brazos, Guadalupe, San Antonio, and Nueces River basins. All these locations were previously sampled by Edwards in 1980. Sampling is done annually using multiple pass electrofishing, and the stream gauge data is downloaded from the USGS gauges and analyzed using indicators of hydrological alteration. We also then run the principal component analysis to find the output of annual flow metrics for each station. And so the otoliths and scales are analyzed for each of the individual samples and then related to that nearest stream gauge. Lengths will then be back calculated from otoliths using the direct proportion method. So we have run into problems with low sample size for our range-wide collection. Population of Guadalupe bass definitely seem to be centered at in the Colorado River Basin, and so we're having difficulty then catching them outside of that. And in 2015, after even visiting the nearly 100 sites, we caught 21 individuals, and in 2016, our field season came to 62. We did incorporate some angling in during our 2016 field season. You can see this age distribution chart shows that we also need to represent a sample from those higher age classes, as that will then give us data with the back calculated growth for each of those years that those squads were growing. So the reason the Colorado River is not included, the Colorado River Basin is not included, is because that research is already being done by Jessica Pease as a part of an ongoing study for Texas Parks and Wildlife. But the final report would then include data collected from the Colorado River Basin and these other 4 river basins as well. But just the issue of then incorporating, of getting our sample numbers high enough within these river basins. So now here's where you can help. We have a call out to other agencies, anyone working in these systems, that if it is within their protocol to do so, to collect Guadalupe bass so we can run these age analysis on them. So basins where samples are needed include the Guadalupe River, which including both the North Fork, South Fork, Johnson Creek, the San Marcos River, and the Blanco River. We also need samples from the San Antonio River Basin, including the Medina River, Cibolo Creek, and Leon Creek. San Antonio River Basin is a site where we have not found any Guadalupe bass during our 2 sampling summer so far. The Brazos River Basin, we also need representative samples. This includes both San Gabriel, the North and South San Gabriel, Brushy Creek, Hog Creek, and the Lampasas River. We also need samples from the Nueces River Basin, including the Sabino River and Frio River. So protocol for our collection of Guadalupe bass is that they're sampled within the basins of interest in a relative proximity to a USGS stream gauge. We are targeting adults from previous research and what we've seen with age analysis. That means like they're greater than 100 millimeters. The only data really needed is the sample location and sample date. Freezer storage of whole fish is preferred, and we can pick them up. But if storage is an issue, then we mostly just need the scales and otoliths, even though we are both analyzing stomach content, and I've been taking morphological pictures of the fish as well. Sampling for our range-wide will continue through November 2017. So that's all I have for now. The goal right now is to increase the sample size of the range-wide and continue getting my metrics in for back calculated growth and relating that to the flow of fish. This research has a potential to tell a lot as far as environmental flows and the flow ecology of this fish throughout their range. So, yeah. So I could take any questions at this time.
I'm going to ask the folks who are on the line, if you have a question, to go ahead and use the little raise hand button And as you're doing that, I'm also going to, we have a very small group, so I'm just going to unmute people. I'm unmuting Blair and Matt and Scott Alford. We have 2 other people that are viewing but don't seem to be connected. So everybody is now unmuted if you'd like to ask any questions.
Yeah, Greg, it looks like Steve McNally has his hand up. Steve, you may have to be muted on your end. While we're waiting on Steve, Heather, I want to thank you for presenting an update on the project, and I understand that you had a call with some folks at Texas Parks and Wildlife Department last week about trying to augment sampling in those identified reaches. Do you get a sense on at this point what sort of collaboration you may have with folks in the Inland Fisheries Division?
So I'm really not sure at this point then on what connections are feasible and able to be made. So I mean, my plan is to start just like reaching out more to make those plans as the spring season comes, because I will be out in the spring for collection.
Okay. Matt, I think you were on that call as well. Is that right?
Yeah, can you hear me?
Yes, I can.
Well, and we talked about some targets that Heather was shooting for, and the Colorado River's adequate, but there are resources there at Texas Parks and Wildlife and the Inland Fisheries Division there that will help, and if we could schedule that ahead of time when Heather knows she's available and she's going to be out collecting, our folks with TPWD could be available, but just needs to be scheduled. And there was an offer to do that at Steve's. It's just a matter of communicating that.
Okay. Was there talk about how much lead time? I know more is better, but if y'all are scheduling to go out at a certain time, how much headway or lead time you'd like to have?
Well, if Steve could answer that, that would help.
Okay. So we have a question here from Steve that says, Heather, how are you handling reaches where there's a high level of introgression with smallmouth bass, such as the San Marcos River?
That is a good question, and I do have like indicators like telling me where they are hybridized. I haven't, from what I've seen, encountered any pure smallmouth. Some may be hybrids, but I can't tell. I always identify them down and key them out as Guadalupe Bass. But I have been taking fin tissue samples from all the fish I collect, including both young of year on the North and South Lano River, and then all of my range-wide fish. I always take the fin tissue sample. And I am interested to run that genetics if I find the means, or if any other organization is interested in running those fin clips that I've collected. It would definitely be interesting to know any hybridization rates, because I could then continue with ongoing analysis of that as well.
Can you hear me now?
Yeah, hi.
Oh, hi. I had a problem, a technical problem here with my phone. But yeah, what I'm wondering about is, in terms of the range-wide assessment, we did a range-wide assessment looking at Guadalupe Bass genetics. Are you trying to concentrate, on the growth and recruitment stuff in areas where we have?
Where you have what?
Low levels of integration. And in other words, where we don't have a lot of hybridization. The North and South Lano are the level of integration there is really low. But San Marcos, I believe, I have to go back and look at the publication, but it's really high. So what you're what you're looking at there are fish that are mainly smallmouth, and you know, I'm wondering how that fits into, you know, any type of recruitment hypothesis when the fish you're looking at really isn't the Guadalupe bass.
Exactly. So we do target areas that have low rates of integration, but and then we'll just like relate that in if we like from fish samples in these places. So it's like not just the San Marcos River. I guess the Lampasas is in that in the Blanco as well. So we hope to run the genetics on it to be able to incorporate that into our recruitment, because obviously we can't say much about recruitment of them if it's not the species we're targeting. Yeah.
Yeah, the Blanco. I mean, if you get below the Narrows, it's very, very highly hybridized with smallmouth. But the area above the Narrows now is, since we did a repatriation project, is almost 100% Guadalupe bass. It is 100% Guadalupe bass. So, anyways, that's just something I'm wondering how that's going to be parsed out. You've answered my question. That's the only question I had.
Okay, Heather, I know from talking with a few folks from Texas Parks and Wildlife Department that there may be opportunities to engage with fly fisher groups in health to augment the sampling, and I'm wondering specifically how you might try to reach out to those groups to encourage contributing to those samples.
That's a good question, because it could be a potential resource. I hope to talk with Dr. Gleason about that, because I know that he works with angling groups quite often, especially the fly fishers. But my only issue with that, and what I'm not sure how to bring up, is that we're collecting these fish for otoliths, and so they do have to be sacrificed. And a lot of these groups are very adamantly catch and release, and it's hard to then relate to them to let them know like what we're doing and like why the fish needs to be sacrificed, and like the data that we're then finding from it. I feel like there's some disconnect between that. So that's my issue with relating to anglers. But if people think that would still be a good idea, I'm definitely willing to try.
I understand the potential disconnect there, but you know that anglers are primarily concerned with the resource, and I'm sure you could craft a message that, you know, by contributing specimens collected from these reaches, you're informing, you know, information that will lead to a better understanding of the population and hopefully influence management of those resources. So I think there's a message there to encourage the stewardship while at the same time having to sacrifice individual fish. So,
great. I agree.
Just really would encourage you to continue talking with Dacus Kiesling about that. So, are there any other questions at this time for Heather? If not, we are going to. This is being recorded and posted, and I encourage anyone who has questions and interest in this project, and also the population of Guadalupe bass and how you might better connect with those efforts. Both Heather's email address and Dr. Verbal Pearson's email address are on the screen, and we just encourage you to contact them directly. And also just really encourage you, Heather, to continue reaching out with the Texas Parks and Wildlife Department primarily, and others to ensure that the sample size for this project is good for reducing uncertainty in estimates for statistical purposes. So,
yeah.
Also just want to thank you again for coming on and presenting solo for the first time. So really appreciate that. So, that concludes the December 2016 webinar. We're not going to have one in January, but we will have one in February. So be looking for an announcement from us I'll just do a last call for questions or comments. All right. Well, you'll have a safe and happy, enjoyable holiday season, everyone, and we'll see you in 2017.
Thanks, y'all.